So my lab is interested in the development of new high-speed volumetric imaging techniques with the goal of understanding how neuronal circuits give rise to behavior. And you have heard already a number of fascinating talks today and also the motivations for why we need these tools. For us, I just want to briefly summarize also why, from a biological point of view, we are developing these techniques. And this is basically comes down to what has been already mentioned previously, which is related to the incredible density of the interconnectedness of the neurons in the brain. So if we think about the human brain that has about 80 billion neurons and we take out 2 neurons randomly, they are only connected over 2 or 3 synapses. And so this alone shows us that in many cases information may be not represented by the individual elements of the systems, but rather by states of the network, and therefore we intrinsically need to move forward and record from many neurons at the same time. So these are some of the questions that we are interested in approaching by developing these technologies in the lab. And you have heard also about parallel efforts that are going on the level of connectomics that are related to understanding the wiring diagram or the connectivity. But I think connectome will not be answering us these questions. It may be, so the connectome may be putting us a constraint on how we could approach this question, but it will be not sufficient to answer them. And the best example comes from C. elegans that we also heard about. The C. elegans had only 300 neurons. The connectome is mapped out already for over 30 years. Nevertheless, we still cannot predict the behavior of C. elegans. So that is really the motivation for us, why we want to move forward and develop move towards functional maps of neuronal circuits. And in particular, what is here necessary is we need to have tools that allow us to do 2 things. On one hand, we want to be able to do stimulation of defined spatial-temporal patterns of activity on a genetically identical set of neurons, and combine that with a parallel and unbiased recording from an entire population while we are doing that. And you can also think about, if you had these tools, how we would be going conceptually about approaching some of these questions. And what we would like to do is we would like to use methods from machine learning, graph theory, and information theory to approach them. So in this example, you can imagine that the first set of tools could allow us to position ourselves in each of these individual states of this abstract space that is the state space. And then by having the ability to observe how these points evolve to other points, we could build over time transition probability maps that could inform us about the probability of the transitions and use them to inform models that could be generative of the dynamics of the brain. And ultimately, what we are interested in to work towards the discovery of the underlying algorithms. So about 7-8 years ago, when we started with this project, we first focused on the first topic and developed, basically based on optogenetics, methods that allow us to sculpt the distribution of light and thereby stimulate individual neurons at high speed and resolution. And more over the last few years, we have focused on the second topic, which is the parallel and unbiased recording. And here we have developed a portfolio of different techniques that are addressing this question from the level of small organisms on the C. elegans, and more recently in the more scattering brain of rodents, and we are moving also towards the primate imaging. And today I would like basically to give you a vignette of some of the techniques that we have developed, mainly around the idea of light sculpting that has found application both in the smaller semi-transparent C. elegans, but as I will show you also could be a useful technique for imaging the rodent brain. So I would like to introduce the basic idea of light sculpting, and those of you who are familiar with optics and microscopy techniques quickly realize that in any kind of microscope, basically, the 2 parameters that are describing the confinement of the light in our sample are given by 2 things. One is the spot size of a beam that we choose to illuminate the sample with, and the other one is the depth of field, or the axial confinement. And you have heard this also in many talks. And the problems that we are facing in some sense is the fact that these 2 parameters that are fundamentally defining the properties of light in the sample are both given by the wavelength of light, lambda, and the numerical aperture of the optics, which is basically the ratio between the diameter of the lens and the focal length. So as such, the two are inherently coupled, and so whenever we choose our spot size to be a certain size, we inevitably end up with a certain axial confinement or depth of field. And what we are basically asking is how can we go beyond that and come up with a technique that can allow us to create more arbitrary distributions of light within the sample. Now, as you have heard, the lambda is basically the wavelength of light. So if we set also our wavelength fix, then there is no way around this coupling. So we will end up inevitably for every given spot size with a given depth of field. However, if we go to a different situation, you heard also about two-photon microscopy. And in two-photon microscopy, this is typically based on the use of femtosecond pulses of light. And due to the so-called uncertainty relationship, which says that if you choose the time very short, you end up with an uncertainty in energy, which translates in an uncertainty into colors, means that if we use femtosecond pulses, we are no longer dealing with a single wavelength, but with a spectrum. And so you can see at least that by exploiting that spectrum effectively, this constraint that I was mentioning before could be relaxed. And so one way how this can be done has been shown to be through the technique of so-called temporal focusing that I would like to introduce to you briefly. So the basic idea is we use these femtosecond pulses of light. We send them to a dispersive element, for instance, a grating. And what the grating does is it sends the different spectral component within the pulse along different spatial directions. So as the pulse is propagating away from the grating, the peak intensity drops off as a function of propagation, and this leads basically to a dilution of the photons in space. So a pulse like that is not very effective in exciting the molecules. So we reduce the two-photon excitation probability as a function of propagation. Now we reverse the process by using a telescope that is consisting of a lens and our microscope objective. And what that telescope does is it forms an image here in the sample of our originally short pulse on the grating. So what that means is that there will be only one plane in the axial location where all these spectral components will be again co-localized both in time and space, leading to this high energetic pulse that is very effective in exciting the molecules. But outside of that region, we are basically dealing with a dispersed pulse, and such pulses are not very effective in exciting the molecules. So what we can achieve in this way, we can achieve the localization of excitation in the axial direction by manipulating the spectral properties of the pulse and thereby basically decouple it from the lateral confinement and reserve the lateral confinement to basically choose the spot size that we are interested in the sample by basically illuminating the pulse on the grating with a different size. Just to draw the comparison to the standard two-photon microscope that you are more familiar with, in the standard two-photon microscope, both the localization laterally and axially are generated by the focusing in space, and in the temporal focusing, the axial localization is generated by manipulating the spectral properties of the pulse, and laterally we can focus in space. So going to the problem of large-scale volumetric imaging, we thought instead of now exciting each of the neurons at once and scanning it, we could use that to create a sheet of light, similar to Phillips light sheet microscopy. In this case, we can create a sheet that is spanning over about 75 microns, but still be actually confined to about 2 microns. So the main difference here is that it's a two-photon excitation, and it doesn't require the geometry that you saw before, where you have the two objectives being orthogonal to each other. So the idea was exciting a field of view of that size and then scanning that sheet axially quickly to capture the volume. And we wanted to use GCamp again as our calcium indicator. And the reason why we chose that size was that we were interested in looking at the brain of C. elegans that is roughly would fit in that volume. So now if you want to do two-photon excitation of a sheet of that size, this doesn't work with the conventional laser sources that people have been using for two-photon scanning microscopy. You need significantly higher pulse energies. And for that, we used a so-called regenerative amplifier that is shown here. Basically, it is seeded by a conventional two-photon laser, And what it does, it creates significantly higher pulse energies, but at the lower repetition rates. And so the pulses are now sent to the grating, and here you have the temporally focusing setup that I was describing. And the fluorescence is now excited here and then collected, separated by a dichroic, and sent to a detector that we have designed based on a so-called image intensifier and a camera. So overall, you can see that we can create now a sheet that is about 75 microns and being axially confined to 2. And what this allows us basically is to go at very high speeds, about 200 frames per second, or video rate volumetric imaging. So as I said earlier, we wanted to apply that to the brain of C. elegans. And the neurons we wanted to record from here are located in so-called head ganglion that are shown here. And this was done in a collaboration with my former colleague Manuel Zimmer, who is at IMP. So when we first expressed GCAMP into all of these head ganglion neurons, we saw that due to the incredible density of the neurons and of the small size, we couldn't distinguish them individually. So we came up with the idea of using a nuclear localized version of GCAMP. Basically, it has a nuclear localization sequence that drove the expression of GCAMP into the nuclei, and then we could nicely see that the neurons were localized in all dimensions. So we could apply our wide field imaging technique, and what I'm showing you here is now a three-dimensional maximal intensity projection of about 120 neurons of the worm, and during spontaneous activity. So you see significant spontaneous activity. Certain neurons are correlated, others are anti-correlated to each other, and this is all recorded at about 5 volumes per second. Our method can go about 4 or 5 times faster, but neurons in C. elegans have intrinsically low responding or slow responding time scales, so we chose to image them at the lower frame rates. Now, if you have such a high dimensional system and you want to make sense out of it, so one simple way of asking what are these neurons telling us and what are they doing is to do dimensionality reduction. And one simple way of doing it is to do principal component analysis, which is like asking what are the most components in this system that it give rise to the variance that we are observing here. So when we do PCA, we observe that about 70% of the variance of this data was explainable by the first 3 principal components that are now shown here. And here you see the color is now encoding for the time. Now again, the great advantage of C. elegans comes from the fact that we have the connectome, so we can map these principal components back to the known connectome. And what we found was that the first principal components was basically an antagonism between different interneurons and head motor neurons. The second principal component was a class of head motor neurons, and the third principal component was antagonism between different head motor neurons. And we could now display these principal components in the phase space, and what we observed was an interesting behavior which showed sort of a trajectory that was reproducible and also was characterized by regions that resembled attractors, which you can see now in this movie much better. As the states of the brain are evolving, you see that the brain seemed to be spending time in one of these attractors, and then it's switching back and going to another attractor, coming back again, and going on and so on. So this was an interesting observation, and now we have increasing evidence, mainly from the lab of Manuel, who has been looking also in these animals during free behavior. And what we know is these attractors correspond now to unique motor programs, such as forward crawling, backward crawling, left and right movement, and so on. So it seems that during spontaneous activity, the brain is randomly switching between these possible motor programs. And in the presence of the stimulus, this landscape could be tilted such that the probability of one of them being assumed over the other could be modulated. So we are following up on this biologically and interested in now developing techniques that allow us to observe this behavior during free navigation as the animal is making left-right type of decisions, and see what kind of things can we observe here. So while working on this, we have been also trying to push these methods further, and I would like to give you an overview of another technique, and this is related to how can we extend these techniques into the mouse brain and look at the larger scale in scattering tissue. So since many of you are familiar with conventional two-photon scanning microscopy, I just want to briefly take a moment and outline the limitations, the fundamental limitations of two-photon microscopy. Let's assume that we were interested in recording from a volume that is about 500 micron cube. And this would be about the size of our diffraction limited spot. And we want to record from this volume at about 5 hertz. Now, if we do the simple math, these are 500 million voxels, and recording them at 5 hertz requires a 2.5 gigahertz acquisition rate. And therefore, if we want to at least excite each pixel with one pulse, we would need to have a laser that runs at that repetition rate. So lasers are conventionally not available at this rate, but even if they would be, we are facing another problem, that is that each individual voxel we will be spending only 0.4 nanoseconds, which is about an order of magnitude shorter than the fluorescence lifetime. It means we will not be able to collect any fluorescence out of these voxels. So one conceptual solution that we could employ here is to minimize the spatial resolution or the sampling rate to the limit of the structure of interest. So if we are interested, for instance, in recording from individual cell bodies or from individual neurons, this resolution in some way may be too good, and we are paying the price for that by having a reduced volumetric acquisition rate. So the question becomes, in some way, how can we relax the spatial resolution in a meaningful manner? So cortical neurons are on the order of about 10 to 15 microns, and this is the size of the point split function. And in some ways, people are mainly using that, at least for these kinds of imaging, because they are mainly interested in the axial sectioning capability. So we could, in principle, relax the spatial resolution laterally by, for instance, underfilling the microscope objective. But if we do that, then we will end up with a beam that is going axially over 70 microns for the reasons I mentioned before. So what we really want is to be able to sculpt an isotropically sculpted point split function laterally and axially. And thereby we would also need to be able to decouple again the lateral from the axial confinement, and this is again where light sculpting comes in. So the idea here is now, instead of sculpting our point split function to be a large sheet, as I was showing you before, in this case we sculpt it to be an isotropically shaped 5 micron sphere, which is shown here, and then we can scan this by various methods. This is in conventional way, laterally and axially, to cover the volume. So going back to the example that I showed you, so the 1 million pixels are now translating acquired at 5 hertz to about 5 megahertz acquisition rate, and which is more realistically possible with laser technology, and also we get a much higher pixel dwell time at each of them, so we can collect significantly more fluorescence. So nevertheless, if you look at the size of this point split function, it's still 130 times larger than the conventional diffraction limited point split function. So lasers that were developed for that excitation wouldn't work here because they lack, again, the sufficient laser pulse energies. So we built a laser in our own lab that was running at about 4.1 megahertz and delivered 0.5 microjoule pulses that are enough to excite these sized point split functions. And this was integrated into a scanned version of the temporal focusing setup, which is consisting basically of mirrors and that are then scanning the beam that is illuminating the grating, and the grating together with the lens and the objective is performing the temporal focusing, and we are scanning it up and down with an objective, piezo. So we collect now the fluorescence instead of on a camera from each of these point split function on a PMT, and the acquisition is also synchronized with the excitation. So we are running in the domain that we have in 1 pulse per pixel only. So this is just an impression of the home-built laser and its integration to the scan temporal focusing microscope. Next, we were interested in recording the activity from in vivo in the mouse brain. So we used mice where we expressed a GCamp in the motor cortical areas and the PPC while they were awake and were running. So with this, we can now go deep and look at the layer 2 tree neurons and record from them, as you can see here, at about 200 microns depth. from about 110 neurons or so. We can go also deeper to layer 5 neurons at about 470 microns, record from a similar number of neurons. And what is important here now to realize is we are now looking at a field of view that is about 500 by 500 and can capture that at 160 hertz. So this is way too fast for a single plane given the slow response time of calcium. So what we could do is to use the time instead and look at 2 planes, for instance, at the same time, which is now shown here, one at 150, the other one at 350 microns, recorded simultaneously. Or going back to the example that I was outlining, we can now go here and record from a volume that is now about 500 micron cubed, encompassing about 4,000 neurons, and record all of that at about 3 hertz volumetric rate. So here you start also to see this layered structure of the cortex, the layer 2, 3 neurons and the layer 4 neurons that seem to coming out here. And one of the things that we are now collaborating with other labs is trying to understand basically how the cortex performs computation. So we know that computation arises from complicated interactions between different layers of the cortical neurons, but how that is exactly done is not very clear. So just to show you what we can also observe if we do some simple data analysis, this is basically looking at the top 0.1% cross correlations within a given time window. What we observe are also interesting functional connectivities or networks that are forming transiently, then changing over time. Here the strength of these lines are indicating the degree of the correlation. So one could look at these things in principle and ask how do they change under different behavioral states on the diseased states, and so on. So just want to briefly summarize. So we saw that this idea of light sculpting is possible by decoupling the lateral from the axial parameters of the beam. And this allows us in the wide field configuration is useful for recording at high speed in semi-transparent organisms like C. elegans. And in combination with scanning, it can be a method that allows us to record on the single cell resolution from a large number of neurons in the scattering brain. Finally, I just want to thank the people in the lab who have been involved in doing the work, and our collaborators, as well as our funding sources. Thank you.